the store is incredibly dope and incredibly cool.
It's a bit sad to give this talk and realize that there will be no lobster dinner afterwards to enjoy. But we're of course very grateful to the staff that this meeting does take place so efficiently. So what I wanted them to start by doing is simply remind you about what you all already know, I think, that modern humans evolved in Africa. And sometime less than 100,000 years ago, maybe around 70,000 years ago or so, they start spreading seriously out of Africa and the Middle East and meet and replace other forms of humans that lived in Eurasia, most famously then Neanderthals in Western Eurasia, and other forms of now extinct hominins in Eastern Eurasia. And our lab is then obsessed, if you like, with Neanderthals. And to the left here is a Neanderthal skeleton. So these robust hominins. And they appear in the fossil record of Europe and Western Asia around 400,000 years ago and disappear around 40,000 years ago when modern humans on the right here appear. And we are so obsessed with Neanderthals basically because they are the closest evolutionary relative of all present-day humans. So if we want to define ourselves from a genetic or physiological perspective as a group, it's really them that we should compare ourselves to. It's also interesting that they were here rather recently, just 40,000 years ago or so. So it's interesting how they related to modern humans when they appear. So if you then want to study the genomes of Neanderthals, you have to deal with some technical issues, very little DNA there. The vast majority of the DNA in fossils that are tens of thousands of years old is microbial. And so even tiny amounts of contamination from present-day humans can distort your results. And an important factor is that those fragments you retrieve from the fossils are very short. This is even from a very good specimen. Much more typical is that you have nothing about 60 or 70 bases in length. So when we then map these fragments to the human genome, an issue is that we can only map the parts of the genome where we can confidently put down and map such short fragments. So in reality, it's around 2/3 of the genome that we have accurately sequenced when we say we sequenced the Neanderthal genome. And already back in 2010, then, when we had the first very rough draft of the Neanderthal genome, it became clear that no matter what present-day humans we looked at outside sub-Saharan Africa, they carry something like 1 or 2% of the genome from Neanderthals, suggesting then that when modern humans started leaving Africa, they early on started mixing with Neanderthals. We now begin to learn much more, of course, primarily because we now have some high-quality Neanderthal genomes. So from this site in southern Siberia, in the Altai Mountains, the Niseva Cave, there is one Neanderthal genome of 50x coverage. There's one from Croatia in southern Europe, 30-fold coverage. There is a genome coming along that is almost 30x coverage from the next valley over, if you like, in the Altai Mountains. That genome you can download, and there's a paper coming out very soon on it. And one interesting thing then when we look at these three genomes is that this more recent genome that's around 80,000 years old or so in southern Siberia is more closely related to the southern European Neanderthal here than to the older Neanderthal that lived some 40,000 years earlier in the next valley over. If you look on more Neanderthal genomes, so more modest quality that we have from Europe, going back in time up to 120,000 years, it turns out that they are actually all more closely related to these Western type Neanderthals than to this old one from the Altai Mountains. So this then suggests that sometime between 120,000 years ago and 80,000 years ago, there was a movement of Neanderthals from west to east, replacing Neanderthal populations in the east. So we're beginning to learn about Neanderthal population history and see that there were actually major events in their history. And we will for sure be learning more about that then in the next few years. What we also begin to learn about from studying these high quality genomes is a bit about the demography of Neanderthals. A striking feature in their genomes is that if we look at the Neanderthal genomes here, that they have these large segments of homozygosity, where both chromosomes are identical, suggesting that the parents were rather closely related, suggesting that they lived in small populations. So you see much more of these homozygous stretches than you see in any present-day human populations. So if you look at these three genomes, we can then model this and show that the two eastern Neanderthals, given some reasonable migration rates between subpopulations, were really very small populations, in the order of 50 individuals or so. Whereas the one western Neanderthal, where we have a good genome, is bigger, although not as big as most modern human populations were. So as now more high quality genomes will be coming along in the next year or two, there will be two genomes from the Russian Caucasus, one 40,000, one about 70,000 years old. There will be a genome from Poland, one from Belgium, and another Neanderthal genome from the Denisova Cave. It will be possible to see if this is a pattern that will hold up. Maybe that the Neanderthal populations in the west were bigger, and these ones on the eastern margin of the range may have been smaller in size. There are, of course, also a number of more modest quality Neanderthal genomes. There are 5 published to date. There are many more coming along. Among them, 61 Neanderthals, where we have captured 1.2 million SNPs from their genomes that vary among Neanderthals and among humans to reconstruct their population history, and that will then be coming along in the next year or so. The other interesting aspect is, of course, comes from then the Denisova Cave in the Altai Mountains, where there was this very small bone where we were able some years ago to reconstruct a high quality genome, and we're very surprised to find that this was not a Neanderthal, although there were Neanderthals in this cave. It was something else that went back to a common ancestor shared with Neanderthals in the order of 400,000 years ago or so. So we then called this group of hominins Denisovans after this site. We have the unfortunate situation that we have Neanderthals from the Denisova Cave, and we have Denisovans from the Denisova Cave that we sort of have to keep straight when we talk about them. And these Denisovans then have contributed to present-day people in Asia. So all over mainland Asia, there is a small amount of contribution from Denisovans, about tenfold less than the Neanderthal contribution to non-Africans. And out in Oceania, there's a much bigger contribution, something like 5 or 6%, so even like 10 times more than the Neanderthal contribution. So Browning et al., together with Josh Akey and his group, has then studied this contribution from the Denisovans and done beautiful work where they compared, for example, in Papua New Guinea here, the segments in the present-day Papuan genomes, how they relate to the Neanderthal genome, and find this contribution that's as close to that genome. And they find the Denisovan contribution that's, however, quite distant from the Denisovan genome we have sequenced. And it's the same situation if you look in Pakistan or India. However, if you look in East Asia, in China or in Japan, you find this Neanderthal contribution. You find this contribution that's quite distant from the Denisovan genome, and in addition, a contribution from some population that's quite close to the Denisovan genome that has been sequenced. So this suggests that there is at least 2 Denisovan populations that have contributed to Asians, and there is some indication that there might even have been more. So I think the picture that's emerging is that Denisovans were probably even much more numerous than Neanderthals and had a more interesting population history that will be revealed in the future with different distinct populations, whereas Neanderthals look quite homogeneous actually. So far, the Denisovan remains have only been retrieved from this site, the Denisova cave in southern Siberia. But so it was very exciting last year when a mandible was described from a site in Tibet, high up about 3,000 meters on the Tibetan plateau. It's a mandible that is quite old, over 160,000 years old. There was no DNA retrieved from it, but with mass spectrometry, a group was able to study some bone proteins, and there was one amino acid there that matches the Denisovan genome rather than Neanderthal and modern human genomes. So it may very well be that this is indeed a Denisovan, suggesting then that Denisovans may have lived in Tibet over quite a long time at high altitudes, maybe even adapting to life at high altitude. Hopefully one will learn more about this from more genomes. At least from the Denisova cave, there are more genomes coming. There are upgrades of some of the previous specimens we have studied, where we now with better techniques can get more of the genomes out. So there will be a 5x genome, a 3x genome of a specimen that is a lot older than these other specimens. And there's also a new specimen that will come along. I wanted to spend 2-3 minutes pointing out a sort of interesting quirk with the genomes of Neanderthals and Denisovans. And that is that when we look at the nuclear genome, we have now seen them, as we discussed, that Denisovans are the sister group of Neanderthals and modern humans fall outside them. However, if you look at the mitochondrial genome, the picture is different. There, the Denisovan mitochondrial genome falls outside the variation of Neanderthal mitochondria and modern human mitochondria. So modern humans and Neanderthals look like sisters. So there is a discrepancy here between the mitochondrial genome and the nuclear genome. There is another observation of relevance for this, and that comes from the very oldest hominin specimens from which DNA has been retrieved. It's a site in Spain, a bit over 400,000 years old, where it's a deep site where Matthias Meyer in our department has been able to retrieve a little bit of nuclear DNA from some of the individuals that have been found there. And these individuals are clearly early Neanderthals, are on the Neanderthal lineage. However, when he sequenced the mitochondrial genome from this individual, or from one individual there, it fell together with the mitochondrial genome of the Denisovans and falling outside as an outgroup to Neanderthals and modern humans, although this is an early Neanderthal or an ancestor of Neanderthals. So this led us and others to suggest that maybe there had been some gene flow into Neanderthals from Africa, ancestors of modern humans, that introduced this mitochondrial DNA to Neanderthals. So it would of course be very interesting to look not only on the maternal inherited part of the genome, but the paternal inherited Y chromosome also. And over the last few years, it has actually been sort of a frustration to us that all the Neanderthal genomes from which we have retrieved reasonable amounts of DNA have turned out to be female. We have not had a chance to look at the Y chromosome. But now Mattea Heidenjak have looked systematically on many specimens and identified 3 Neanderthals, male Neanderthals, for which you can retrieve DNA, and 2 denisovans of the denisovans from the Denisova cave. So Martin Teterer then designed, together with Janet Kelso, her group have designed capture probes for about 7 megabases of single copy DNA on the Y chromosome. So we can now have a first view of the Y chromosome of both Neanderthals and Denisovans, and they have sort of reasonable coverage for these. So if we then reconstruct the phylogeny of the Y chromosome, the Denisovan Y chromosome again falls as an outgroup to the Y chromosomes of Neanderthals and modern humans. So it looks like the mitochondria. So this then supports a view where Neanderthals are the sister group of Denisovans, but that there have been some gene flow from ancestors of modern humans in Africa into Neanderthals that then introduce the Y chromosome into Neanderthals and introduce the mitochondria into Neanderthals, so that late Neanderthals have this sort of Y and mitochondria more related to modern humans. One can wonder why that would happen, why would that replacement take place? And I think there are sort of indications from several studies that the effective population size of Neanderthals was so much smaller than of modern humans that slightly deleterious variants tended to become fixed among Neanderthals. And that may particularly affected sort of non-recombining parts of the genome, such as the mitochondria and the Y chromosome. So you can show by modeling with reasonable population sizes that the selective advantage of the modern human Y or mitochondria of say 2% would actually result in a 50% chance that they became fixed in Neanderthal. So this then supports sort of other lines of evidence that there was gene flow into Neanderthals that didn't affect the Denisovans from modern human ancestors. That was presented also 2 days ago at this meeting run by David Harris, for example, from the Tishcon lab. What is also happening, I should say, is that several of these other gene flow events that we infer from the genomes, we begin to see direct evidence of it from DNA from fossils. The first case of that came a few years ago already, which was in Romania, this mandible of a modern human that's around 40,000 years old. So when we looked at the genome of this individual, we found 7 large chromosomal segments that is purely of Neanderthal origin. So this then suggests that this individual had a close relative who was Neanderthal, and you can show that 6, 5, or 4 generations back, this modern human 40,000 years ago in Romania had a Neanderthal ancestor. So we sort of stumble across someone who is quite closely related to this gene flow that's from Neanderthals into modern humans. The other striking such case comes from the Denisova cave, where together with Tom Hyan's group in Oxford, we have screened thousands of bone fragments that we find. The vast majority of them come from animals, but now and again we find a hominin bone fragment. And one of the fragments I found here, when we studied the genome and plot on the chromosomes here in blue alleles that are Neanderthal-like and in red ones that are Denisova one-like, you find that all chromosomes are almost homogeneously both blue and red, suggesting then that this is a first generation offspring of a Neanderthal mother, because the mitochondria is Neanderthal, and a father who was Denisova. And not only that, if there are some regions here, regions of a megabase or more, one is highlighted here, where this individual looks solidly Neanderthal, so both chromosomes look Neanderthal, suggesting then that the father here had some Neanderthals back in his family tree. So among these gene flow events, we stumble across another one here, the direct offspring of a mixture between Neanderthals and Denisovans. So this sort of begins to be seen like we're almost too lucky. We run into this quite often. It suggests, first of all, of course, that these groups are mixed quite often in the cases when they have met. But it's also really striking that among just 6 individuals from the Denisova category have DNA information. One of them turns out to be this direct first-generation offspring of a Neanderthal and a Denisovan. If we think about modern humans for which we have DNA information and that lived so long ago that they had a chance to meet Neanderthals in their lifetime, we have DNA information from just 3 such individuals, one from Western Siberia, one from outside Beijing, and this individual from Romania. 1 out of 3 of these have a close Neanderthal relative. So I think it will be very exciting in the next year or so to try to find more of these very early modern humans and see how they relate to Neanderthals. Because it might be that one aspect, at least, of how Neanderthals or Denisovans disappeared may have been that they were simply assimilated into maybe larger